All right, you may remember this picture from our last video where I showed you the plasma membrane. Well, now we're really going to talk about it because the goal of this video is for you to know how cells get things in and out. So we call the cell membrane the fluid mosaic model because it's kind of like a boat and it kind of flows all around like this. And everything kind of floats in it and on it. So it's just like a little floating device. And so it's like a lake with little proteins and things floating in it. So the phospholipid bilayer is made up of phospholipids, which have a phosphate head and fatty acid tail. The hydrophilic, which means like water, hydro means water, phileo means to like, is the top part, the inside and the outside, or the heads. And they associate with water because they're polar. The inside is nonpolar because they're fatty acids and they do not dissolve in water. So the tails hang out together and the heads are further apart. Because the heads are polar, they have to be away from each other, right? A bilayer is formed when the fatty acid tails associate with each other and the polar heads face the aqueous environment, or water, because polar molecules like water. All right. And this particular way of doing things is going to become important. And you're going to take some candy and you're going to make one of these. And hopefully this will show you this a little bit better. All right. So two important factors. So if the cell membrane is kind of like the top of a lake, the factors that let you know, is it a choppy lake? Is it a smooth lake? Is it frozen? Is lipid composition. So the types of fatty acids and temperature. Okay. So cholesterol is a fatty acid that lives inside plasma membrane. You've got to have some cholesterol, but if you have too much, it gets really rigid. Okay. If you have too little, it gets too fluidy and it lets too many things in. So you got to have just the right amount of cholesterol in your, plasma membrane lake. Temperature, if you have increased temperature, then usually you will have the ability to get things into the cell a little bit better. However, if it gets cold, it's going to kind of crystallize and you're not going to be able to get anything in or out. All right. We also have these membranes that live there. We have some that live just at the top, some that live just at the bottom, and some that go all the way through. If you live at the top or the bottom, they're usually polar, which means they can't get through this hydrophobic middle or this nonpolar middle. We call those peripheral. Peri means around, so they're around the outside of the cell membrane. Or integral membrane proteins, they go all the way through. You also have these different little signaling devices that we use. A glycoprotein, glyco means sugar, so it's a sugar and a protein, or a glycolipid, a sugar and a lipid. And these are used mostly for signaling, like in our bodies, we have a lot of these, and the signal says, I'm Ms. Klug cell, please don't kill me. So there's a lot of signals out there on the outside of these cells. Now, we talked about selectively permeable. So a plasma membrane is going to let some things in and other things not. And so in order to get things in and out of the cell, it's important that we understand this permeability. So there's two processes by which we can get things in and out of a cell. Passive transport, which does not require energy, and active transport, which requires energy. So if you think about being active requires energy. So passive transport can come in two main types, simple diffusion or facilitated diffusion. Simple diffusion is just going straight through the bilayer. Facilitated diffusion is using those proteins that we talked about a minute ago. Facilitated, if I facilitate your meeting another person, that means I help. So facilitated diffusion is helping something get in and out of the cell using a protein. All right, now let's talk about a concentration gradient. So things move with the concentration gradient. The greater the concentration gradient, the faster substance will diffuse. So in this instance right here, here's the plasma membrane. You have a whole bunch of concentration of things outside the cell and nothing inside. Okay, so you've created a situation where you have lots up here and none in here. What will happen is cells don't like this. They want things to be equal. So they're going to allow in as much of these solutes, as much as these things on the outside, they're going to flow into the cell so that things can be equal. Okay, if you have higher concentration outside the cell, it causes the solute to diffuse out. Solute is the thing being dissolved. And the higher concentration outside causes a solute to diffuse in. Diffusion through a permeable membrane follows a concentration gradient. And I always say it's like going down a slide. At the top of the slide, you have a high concentration gradient, and it's very easy for you to sit on top of the slide and slide all the way down, right? So that's what these molecules are doing. They're at the top of the slide, all concentrated on the outside, and they're sliding down the slide where they're not very concentrated on the inside. And that does not require energy. So diffusion, by definition, is moving of a substance from an area of high concentration to low concentration. Simple diffusion takes place through the phospholipid bilayer, as a molecule that is hydrophobic and soluble in lipids can pass through the membrane. So think about this, guys. Non-polar molecules can pass through without any help. If you are polar, you're going to need some help. Okay? Polar molecules cannot pass through because what happens is they get attracted to the outside of the cell and just kind of hang out there. They can't go all the way through. But nonpolar are not attracted to the outside of the cell and just slide right on through. Okay? Osmosis is specific to the diffusion of water. So this is water moving from an area of high concentration to low concentration. So again, here you have a semi-permeable membrane, you have a solute right here. In order to even things out, the water is going to move. In this example, these particular solutes are too big to move themselves. We can't move the solutes, but the water is very easy. We can just move the water over and even things out so that the concentration is even. So when we are comparing things in a solution, we have some terms: hyper, hypo, and isotonic. So isotonic is when things are equal on the outside and the inside of the cell. So you have a picture of a red blood cell. This is what they should look like as they are floating around your body. This is a normal red blood cell. The concentration of solutes on the inside and the outside of the cell are equal. Here's your regular plant cell. The same thing. This is what it should normally look like. Now, let's say I drink a whole lot of salt water, okay? Or somebody gives me an IV full of salt water, okay? What's going to happen is the concentration of salt will be so large outside my cells. Water, because salt can't come in because it's charged, the water is just going to rush out of my cell and the cells are going to shrink. And the same thing happens here in the plant cell. You can see where the plant cell membrane is actually pulled away from the plant cell wall. And so the actual cell itself is only this size and the cell wall is just left out here with nothing in between. Okay, and this would be a cell that would sort of, or a plant that would kind of slump over because it needs water. We call that hypertonic. Okay, and hyper means over. So this is over the amount of solutes on the outside, or over the amount of solutes on the inside, basically. So this is the exact opposite. Hypotonic is basically when you create a situation where the solution has a lower solute concentration. So the inside of the cell would be full of salt. Water would be rushing into the cell, and eventually the cell will explode. And here in this plant cell, you can see how big the cells got. I mean, compare this to that. The cells got so big, and in plant cells, they'll push against each other until if something isn't happening, that's called turgor pressure, the pushing of the cell membrane against the cell wall, until eventually a cell will burst, and that's called hypotonic. You will be required to know the difference between hyper, iso, and hypotonic. The concentrations of solutes in the environment determines the direction of osmosis. In organisms with cell walls, the cell wall is going to limit the volume that a cell can take up. So turgor pressure is the internal pressure, just what I was saying, between the cell membrane and the cell wall. When you get so much water, the cell membrane is going to push all these cells over. Eventually, the plant won't be able to take on any more water, and some of the cells might actually burst. Now, facilitated diffusion needs some help. So things like sugar or salt or sodium or potassium, they are charged and they are polar, so they need some help. And typically they'll have just a little protein that they'll just bind to the protein, and the protein will spit them in the other direction. The big situation here is this doesn't require energy because the molecule has to be moving from an area of high concentration to low concentration. So in this picture, the glucose, there's a lot of glucose on the outside, and it's moving to an area of low concentration inside. So the only time that using these proteins does not require energy is if you are moving with a concentration gradient from high concentration to low concentration. Now, sometimes because water's polar, it can't get through the cell just all on its own. Sometimes it hitchhikes. So if it can find a charged ion to bind to, it can actually hitchhike through. And when it does that, it's called an aquaporin. Aqua means water, por means hole or pore. And so there are specific channels that allow a lot of water through. Active transport requires energy because we're moving against the concentration gradient. We're going from an area of low concentration to an area of high concentration. So we're trying to climb up a slide. Okay. And that, if you've ever tried to climb up a slide, takes energy. So we're climbing up a concentration gradient. Okay, the energy source that we use is something called ATP, and in the next unit, we're going to discover how we make the ATP, but just know that ATP is energy, and that's what we use. Active transport is directional because it moves substances against the concentration gradient up the slide from low concentration to high concentration, and usually it needs a carrier protein to do that. So this is a great table for you to learn. It talks about diffusion, simple diffusion, active transport, does it require energy? The force, so the force in any type of regular diffusion is the concentration gradient, but in active transport, remember I said we needed ATP. Now we use a sodium potassium pump. If you're going to take my anatomy class, we're going to talk a lot about the sodium potassium pump because this is how we move our muscles. This is how our nerves move. This is how the sodium potassium pump is the problem when you cramp up, when you get a cramp. This is part of the problem, the sodium potassium pump. You don't have enough, usually it's sodium, to deal with this situation. And so this sodium potassium pump is actually what regulates sodium and potassium levels in our body. All right, now we talked about getting things in and out of the cell. We're usually talking about some ions, some lipids, and things like that. But there's a lot of things that are going to be too big or too charged to pass through. And so we're going to do something called endo or exocytosis. Endo, E-N-D-O means in, C-Y-T-O means cell. So we're going to get things in the cell with endocytosis, E-X-O, exit. We're going to get things out of the cell by exocytosis. So there are 3 types of endocytosis. First of all, phagocytosis, P-H-A-G-O means to eat. And C-Y-T-O means cell. So this is literally cell eating. This is a cell. It sees a bacteria. It gobbles it up. It puts it in a vacuole. And then the lysosomes break it down. Pinocytosis, P-I-N-O means to drink. Cytos, C-Y-T-O means cell. So this is cell drinking where it just takes in liquids or charged particles in water and brings them into the cell in a vesicle. Receptor mediated is when it's only going to be bringing in a specific type of substance. And then it works very similar to phagocytosis in that, but it's only triggered by the specific thing that it's trying to bring in. So this happens sometimes when you're talking about steroids. Steroids use this model. Well, protein steroids. Exocytosis basically is just I have waste, like think of pee and poop from a cell. It gets surrounded, like we have a bladder and then it exits the body, or we have a large intestine and then it exits the body. Well, this is a vesicle. It holds the pee and poo, takes it out to the outside and pushes it out to the rest of the world.